switching gears. So I am here to talk to you about the experimental side of single cell RNA-seq or single nucleus RNA-seq. I do this all day, every day. Is anybody else a wet lab at all? Anybody pipette? Nice, okay, that's actually more than I thought, cool. Has anybody done a single cell experiment before? Great, mostly 10x, I'm assuming? All 10x, cool. Yeah, so that is probably the most popular platform. I'm going to try to touch on some other platforms today, just so that you're aware of them and the people who don't know anything about the experimental side of things can kind of see a broad overview. My first slide's kind of a joke, like why do single cell sequencing? Oh, because you'll get published. This is from 2019, and I just chose like a handful of really interesting papers that use single cell RNA-seq. I'm sure there's so many more now that it's 2021. As Sam said, single cell RNA-seq is just exploding in popularity. New methods are even being developed on the wet lab side, one of which I will go over. So yeah, but actually why do single cell genomics? Individual cells have very distinct biological functions in a complex tissue. There are many subtypes like the immune system. There are how many types of T cells? I don't know. Brain has a ton of different cell types that all have their distinct function. They get impacted differently during disease. They regulate different aspects of the brain. We all know like tumors have a very heterogeneous like microenvironment. Let's see, individual cell types themselves are also very dynamic, such as during differentiation, like from a myoblast to a myotube, from any of these, like from a stem cell to any of these different cell subtypes you can see here. Reprogramming, such as in like iPSCs, like making a regular cell a stem cell or vice versa. So with single cell genomics, we can actually identify and characterize these cell types by their gene expression. So we all know that every cell has the same genome, but they all have different transcriptomes, which lead to their distinct functions. So this is what we're doing with single cell RNA-seq. So just comparing bulk RNA-seq to single cell. Bulk RNA-seq is still, well, kind of fading in popularity, but it's still useful in many ways. You can still say, look for like disease biomarkers. You can, what else do I say this? I had examples, I forget. It's a lot easier to get high quality data out of bulk RNA-seq. The whole process is a lot easier. You can put an entire tissue in lysis buffer and you know you got all of the cell types. Of course, you don't know which reads belong to which one, but bulk RNA-seq is still pretty popular, I think. But everything is pretty much trending to single cell, and not just RNA-seq, but specialized assays like microRNA-seq, long read RNA-seq. We're trying to get to single cell world. Probably others that I don't know of, methylation maybe, I don't know. But yeah, you can kind of see here that like this is an example where each of these colors is like a gene, and when it's in bulk world, it's all averaged out. You have no idea like if one cell type is overexpressing one gene, another one isn't. So you can see that these 4 cell types, they all have different gene expression profiles, and thus you can kind of, like this would be maybe a T-SNE or UMAP, you can kind of see like, yeah, how they kind of cluster together or how they, I don't know, function in relation to each other, you know, using your fancy single cell RNA-seq analysis methods. Single nucleus is very, very, very similar. People will use this in place of single cell RNA-seq when it is just too difficult or is even impossible to do single cell RNA-seq. You are missing obviously all of the cytoplasmic transcripts, so there's some cons associated with that, but overall, it's a pretty robust and reliable way to get some single cell profiles out of your tissue. And this is primarily what I personally have been doing, is single nucleus RNA-seq, rarely single cell actually. Yeah, so for example, I'm going to get to this later, but sometimes single cell RNA-seq is impossible for certain cell types, like neurons and muscle cells are just too big to pass through microfluidics channels of like 10x. You could probably, or even like the FACS sorter will have issues. Sometimes you just can't do single cell. So you're pretty much limited to single nucleus. I think, so I've been working with a lot of flash frozen tissue, and I'm pretty sure, I've never tried actually isolating cells from flash frozen tissue, but I don't know. Nucleus is a lot easier in many ways, I think, like experimentally. No, not quite. It's a little different. It's a little different. But yeah, like with single cell, it's just going to be more, probably more optimization will go into your sample prep for single cell. Yeah, for different tissues. Yeah. I don't have speaker notes. Do I need them? Clearly. Oh, I did on the bottom. Yeah, it's fine. Okay, so yeah, I just have some example platforms here. So 10x is the one I mentioned. It's the most popular. Bio-Rad DD-seq. I thought it was discontinued, but stuff is still online, so this might still be an option. It's very similar to 10x. They have legal battles all the time. Smart-seq 2, this is kind of what I'm calling the plate-based method, but what I mean by that is like you have to get your cells into, each single cell into a well, so you're probably going to use a fax machine, or fluid MC1 is another method to kind of stream cells into individual wells of a plate. Then you can just kind of pretend that like a bulk sample is in each well, and you kind of process the entire 96 well plate, or like the fluid I'm chip, you know, like it was a bulk library. So you get really high quality data out of that. But I mean, 96 well plate, only get 96 cells, pretty bad. And the last one that our lab is using extensively is Split-seq. It's the one that is probably, have any of you heard of Split-seq? Okay, fairly. Yeah, this one is one of the newer methods. It's a startup company out of Seattle. Yeah, I'll go over what the differences there are. So yeah, it's just cartoons of what the droplet-based kind of looks like. So you have your cell or your nucleus. It's my mouse. Yeah, that is kind of encapsulated in this droplet that contains, yes. I mean, I'm good. I'm good. I'm not used to all this stuff. This is weird to be in real life. So yeah, you have your cell encapsulated in a nanodroplet. It also has this, what they call a bead that has these barcode oligos kind of attached to it that will barcode the cell. And then in comparison, we have the plate-based, which I was kind of saying you have just one cell in a well, and you can just directly lyse that cell without worrying about, yeah, like it's already in its own single container. But with Split-seq, the cells or the nuclei will kind of act as their own fixed kind of containers. So I can't say this. I don't have I have a picture later, I guess. But yeah, Split-seq is very different from both of these, but it is in a way a plate-based method since there's no equipment involved. You can see here that for 10x and Bio-Rad, you need some sort of single cell isolator in order to make these nanodroplets, which these can cost money. Sorry, what? Yeah. Yeah, basically these the cells or nuclei are fixed and permeabilized, and then you will load them into a 96 well plate, and the 96 well plate will have these barcodes, a well-specific barcode for each well. And you dump your cell in, and it's permeabilized, right? So it has holes in it, so these adapters or barcodes can kind of come in to the cell or nucleus, and then the reaction also has like the reverse transcriptase and stuff, so it'll be within the actual cell, reverse transcribed, and then there are other ligations afterwards. And eventually you lyse the cells and you continue on. Yeah. Okay, so yeah, here I have some pros and cons for each of these methods. For 10x and Bio-Rad, I kind of grouped them together. So I guess I'm just reading this off. So in comparison, like Smart-seq, they're the plate-based method, you're gonna have a very high read depth per cell. Like you can get a million reads per cell, which you would not get at all with this droplet-based method or Split-seq, but you do have a much higher throughput for the droplet-based and Split-seq. Like I said, with the plate-based, you're kind of stuck with either 96. I think the fluidime goes up to 800 cells in a chip. So if you don't need a whole lot of cells for the question you're trying to answer, that could be a good strategy. The data is really high quality. You're much more likely to capture full-length transcripts from this plate-based method. Yeah, I covered that. Yeah, I guess this 10x is really fascinating, easy to run. I know a lot of places, maybe including here, I'm not sure, you just drop off your samples and they do it for you. That's insane. I'm not used to that at all, but that's great. So I guess it's really fast and easy to run. I personally I've done the DD-seq attack, but not RNA, but probably similar. It's okay. It's fine. Split-seq is a little more hands-on, a little more, what's longer. I'll get to it. I guess I should have maybe mentioned it first, but you can't just have, like, when you load your cells into the Split-seq well, you're loading more than one cell. There's a group of cells. So how do you make sure that each cell gets a unique barcode? Is actually through the splitting and pooling strategy. It's a combinatorial based approach. So you physically have to, in your 96 well plates, like get all the cells, multi-channel, pool all the cells, put them in a tube, spin them down, resuspend them, put them back in a 96 well plate, do your ligation. It's a bit tedious. Yes. Yes. Oh my gosh, really? That is wild. Yeah. That's awesome. I don't want to be that person. Multi-channel all day. We're trying to, we'd like to get it more automated. That's talk for another day. Yeah, here's some cartoons of just like the different kind of, how do I say this, how the reads will look at the end of the day between the droplet and the plate-based. So plate-based, you're going to have it just bought like a normal bulk experiment. You have like your adapters on either end. Both reads will get the cell ID and the gene sequence. There's no UMI, which is kind of a con. But then in the droplet-based, your read one is going to contain the cell ID and the UMI. Like it's all of read one, and just your read two will have your gene sequence. So it's going to have that 3' bias. If it's normally single cell stuff is 3' and based, but I think there's also 5', but it's going to be biased in some way due to this setup here. Oh, unique molecular identifier. I think I get to that. It's kind of a way to measure or to keep track of like PCR duplicates or like doublets, I think, or droplets without any cells. Yes. Okay. And here I have a cost breakdown if anybody cares. And just like the throughput. So Split-seq is pretty good. We get not quite 100,000 cells, never really, but it's kind of comparable to 10x, really high throughput. Price breakdown kind of ends up being similar. I guess Bio-Rad is pretty cheap. I don't know why I don't know. Did they discontinue or not? I don't know. I thought they did, but yeah, they're still selling stuff. And yeah, so costs are pretty much similar across the board. It's just kind of a different protocol, different throughput. So I don't know, they're just kind of things to consider. I'm considering, don't you usually do, it's like 4 samples for a chip and you can do like 8 samples on it? I don't know. It's like, you have like 4 samples, right? And like each sample is 10,000 cells. I don't know, I got this number, the price from an article, and the article said this many cells. I'm trying to be like, that's what, yeah. That's what 10x is telling you. Yeah, this is what the companies are telling us. Yeah. Yeah. Hopefully these prices are something accurate. Oh, and then I found a price for chromium controllers, 35,000, which actually I think is dropped. It used to be like 50,000, so that's nice. No, I have to ask Heidi. We have one, but I don't know how much it is. Yeah, for a while. So yeah, this is kind of like showing you how the droplet-based system works. You have your microparticles, your beads that have these barcodes on them. You combine them with cells in these water-based droplets in an oil emulsion. So if you stream in beads, you stream in cells, they combine in these droplets, and you can actually kind of see the difference between the emulsion and the oil after the single cell isolation. And this part always makes me very nervous, very careful pipetting at this point. Yeah, makes sense. Okay, and then yeah, for plate-based, just how to actually isolate 1 cell per well. Like I mentioned, you can use a fax sorter, fax machine, or this Fluid MC1, which people have used in our lab before, but I'm pretty sure it's not as popular, running or declining in popularity, just because of the throughput of these other droplet-based methods is so much higher. Okay, so here's finally like what Split-seq is. So yeah, like I said, you like the reverse transcription happens inside these actual fixed cells or nuclei. So the first reaction, you load your samples into 48 wells of a 96 well plate, where each of these 48 wells can technically be its own like sample. Say you can multiplex up to 48 samples technically. That makes sense, because each well has this individual barcode, and that barcode can correspond to an actual like a sample. But we tend to do like 24 samples per plate to try to like make sure that we get some recovery for each sample. That makes sense? Yes. For the first one, you want to make sure that you like, for example, say you have like 4 samples, you want to make sure that, you know, sample 1 goes into these 4 wells. You keep track of that. Sample 2 goes into these 4 wells. It is like more than one cell.
Right. But I mean, is there
Yeah.
sorting or
No, well, you like you could, I guess. I don't. But yeah. Oh yeah. Yeah. That would be, I think, more in the sample prep kind of side of things. Right? Like when I'm doing my sample prep, I just get the whole tissue, not lyse the whole tissue. I'll lyse the cell membrane because I'm doing nuclear extraction, but process it uniformly. But you can easily, like, you know, get your magnetic beads, you're isolating your specific cells, and then that can be your sample, if that makes sense. Yeah. Oh yeah, it's formaldehyde based. Yeah. Yeah, technically there's no restriction on cell size. I've always wanted to do like put a huge cell in here and see what happened, like a huge myocyte, but haven't done that yet. Still doing this nuclear prep. But yeah, anyway, yeah, you have your cells in the first 48 wells, and then like I said, you on the multi-channel pipette pool everything, spin, redistribute into a second pool, which now gets another unique barcode is attached to the first one. You can see here. So you have first, you have a second, you have, and then repeat that for a third. Then eventually you actually split up these cells into sublibraries, which actually have another barcode. So there's 4 barcodes total. But if you kind of do the math and you multiply 48 times 96 times 96 times 8, there's actually 3 million possible barcode combinations. So there's not a huge chance that one cell will have, or 2 cells will have the same barcode. That makes sense? Yes. Wait, what? Yeah, no, it's crazy that it works. Yeah, I've had that comment from a PI before. So. Yeah. Obviously you load more than 100,000. So you do need enough starting material, which is always a concern, like especially if you're doing isolation of a specific cell types, like say, for example, getting enough microglia is always a pain to try to put into the system, but yes. What is that? Is that 10x space? I think they're coming out with something similar. Mm-hmm. Yeah, I have a friend that's working on that. I think she was asking us about Split-seq. It's like, oh, 10x is doing everything cool. I got a lot going on, but yeah, it's similar.
But I think it's you still are limited by how much cells you can load on. Yeah, that sense. You're still going to get like 10,000 cells. Yeah, so you can still multiplex your cells. But like,
what's the benefit?
You might not have like enough cells to load.
Got it. That's cool. Nice. Cool. What else am I talking about? Okay. Yeah. So the first step for both 10x and Split-seq is the cell barcoding and reverse transcription. You want to get that RNA to cDNA as quickly as possible. This also adds that unique molecular identifier, or UMI, and the cell barcode, right? You need to know which molecules belong to which cell. So let's see, this kind of is, I think I like this picture. So your mRNA, usually it's poly-A base. So this bead has a primer that will bind to the poly-A tail of your mRNA. And this barcode includes, you can see here, I guess this is, I don't know what this is. I think, is this Split-seq? Okay, yeah, that's why there's 3 barcodes. I think normally it'd just be like 1 barcode, maybe a linker, UMI, And that again is all encapsulated in this read 1, and then your read 2 is going to start capturing the 3' end of the molecule. Yeah, so I think I mentioned this before. Yeah, it's good to identify the PCR duplication. And yeah, obviously all cells need the same barcode. Cool. Yeah, reverse transcription, I'm sure you all know how this works, but I think all these protocols use this like Smart-seq approach where the special TSO will this reverse transcriptase will add these untemplated Z's to the end of the cDNA strand, which will allow this template switching oligo to anchor to these C's and will allow the reverse transcriptase to just switch seamlessly from the RNA to the TSO for second strand synthesis. So in theory, you can get your complete transcript. Well, for the plate base. Again, remember like the UMI is all in read 1, read 2 is only the actual sequence. So yeah. Oh yeah, so finally after your cells are barcoded and reverse transcribed, or molecules are barcoded and reverse transcribed, you can finally break the emulsion. I don't know how, it's a kit, right? So it's just some reagent you add very, very carefully, and you lyse your cells for Split-seq, and then you can kind of proceed to more normal wet lab kind of things, tagmentation, like bulk RNA-seq kind of things, tagmentation, amplification, cleanup with beads, everything is bead cleanup based. I don't know if you all have used them before. Yeah, okay, so I have some technical challenges here. I've mentioned these before. In my opinion, the hardest part of single cell is preparing your single cell suspension or single nucleus suspension. Not to mention whoever is dissecting your mice or cutting up your bits of human tissue. Like I've just been receiving flash frozen tissue, and it's still quite a challenge to process all of this and get it into a clean, nice, no doublet-free single cell suspension. Yeah, you need enough starting material. So again, yeah, if you're isolating a specific cell type, you might have to do that quite a bit in order to get enough to sequence. There's also trial and error. Like for example, we had test tissues to work on for a while just to get it to a single nucleus suspension and like see that it's looking good before we spent $9,000 on this kit. You want to kind of be quick about it. Like you don't want to have your cells or nuclei on ice for hours and hours and hours and hours. You want to, and I think this is the case for all of them, but I'm pretty sure you can fix and freeze for 10x as well. Let's see, does anybody do that? Or they go right from sample prep to the actual sequencing, because that sucks. You can't fix and freeze? Oh my God. Really? Okay, I Googled it and it seemed like it was possible. Always fresh? Okay, wow, that makes it a long day. Long day, that sucks. Uh-huh.
I don't think you can fix.
How do they even survive? Really? Okay, not recommended. Okay.
So I've seen, especially with human samples.
Yeah.
People like freeze them and
Yeah.
then thaw and that's-
Mm-hmm.
But I don't think you can fix.
Okay. Okay, good to know.
Yeah, but not fixing.
Got it. Got it. So it's a very, very, very long first day then.
Yes.
You're preparing your entire sample and then you're doing the first until the safe stopping point or whatever. So for 10x, that's going to be after your reverse transcription, after you break the emulsion, right? And you get it to some point.
It's after you cell sort. So what you do is, so you dissociate,
Uh-huh.
you have your mixture,
Uh-huh.
you cell sort, and you bring that to the genomics expert facility.
Uh-huh.
Then
Oh, they do it for you. Yeah.
I think you do it yourself.
Yeah, of course you can do it yourself. Why would you? Oh, yeah. Yeah, well, okay, I guess it's a pro of Split-seq you can fix. So we do all of our sample prep about a week or so in advance, and then we do the actual experiment whenever we want. The first day takes what, 6 hours or so? Second day is like 8 hours, 6 hours. So it's not the worst, honestly, as long as you can fix and freeze your samples. Yeah. Yeah, I mentioned the cell size thing. Some of these things just aren't impossible. So this is a picture of like a multinucleated muscle cell, you know, that it can't fit. And yeah, most require some kind of specialized equipment. So you got to go to the GHTF or you got to buy your own. But with Split-seq, you don't have to do that. It's nice. Oh yeah, I have pictures recent, like this is yesterday. So I think I mentioned this, but like, yeah, the different tissues will have different sample preps just based on the characteristics of that tissue. Like skeletal muscle has a lot of like connective tissue. Has a lot of crap in it. You can see here. While as brain, they kind of pop out really easily. Another fun fact, I've been doing like a time course based study, and like the younger the mouse, the easier the nuclei just kind of come out and the cleaner they are.
Is that with all the patients?
Yeah, it's pretty much just, there's more crap, there's more debris, which I can't tell you, but this is obviously a DAPI stain. It's just a bunch of non-DAPI stain stuff in here. Yeah. We didn't do any kind of specialized, like, yeah, no cell sorting. No.
Okay, so if you were trying to like isolate nuclei and just like a sample, would you then like sort out all of the other stuff first, since you can't sort out neurons? And just...
Oh, I don't know. That's yeah, that could be a method. Like, have you heard of, like, Miltenyi has a lot of kind of like isolation based stuff like that, where you have kind of magnetic beads that will bind to some surface marker of your cells. You can either do a, I forget what it's called. You can either take away the cells you don't want or retain the cells you don't want and then take everything else out and then flush the cells you want through. Yeah, and then I guess if you wanted to do nuclei version of that, you have to then lyse those cell membranes and then continue.
And so that's all like all of like the isolation procedure.
Yeah, depending on what you want. Like, yeah. Luckily, oh my gosh, I'm so glad that I don't have to do, you know, CD whatever positive, whatever. It's just the whole tissue, dunk it in. But yeah, as far as I know, we do a lot of magnetic bead-based like isolation. Like I don't, but yeah, but people do. Yeah, and then yeah, Miltenyi is actually a really good company for that. They have protocols for a lot of different tissues, like specific. Like this is the brain, this is for adult mouse heart or whatever. This is for, yeah, they have a lot of stuff. It's actually really nice. Oh yeah, and I have just a picture of when you get doublets, which I mean that this will affect your data as well. Oh, I wanted to mention, yeah, this data does not look great. This data, I'm probably gonna look really nice. Like you will see the difference in your data. Yeah, you want to avoid doublets, of course. There's a doublet is when there's more than one nucleus or cell stuck together, so you can see. Yeah. Huh?
Like what is the result in the like?
Oh, I mean, right, you can't tell these apart, right? They get barcoded the same.
Yeah.
So you can't tell these two cells apart from each other. They're all going to have the same. How do you avoid aggregation experimentally? Filtering somewhat kind of helps. I'm not, huh? Wait, what? Sorry. Oh, I have no idea. Does that help? I don't know. I always try to keep filtering and like.
Every tissue has a different protocol. So what you need to do before you sequence is essentially for 10x. I don't know what it is. For 10x, what you do is you go through up until flow cytometry, and you can get an idea of your aggregates and your doublets and things like this from what Liz is showing us. Look at things and see doublets and stuff like that. So you have to optimize the protocol for the platform and for your tissue, right? So like transcription will help something. So it's just tissue stuff.
Yeah, we've been rushed, so it's been more like a, let's make it a computational problem to remove the doublets.
As we will hear better in the quarter, there are computational ways of dealing with that.
Yeah.
Like sort can actually keep the sequence and doublets and so on.
Yeah, flow cytometry is kind of gold standard. I would love to be doing that in the lab, or at least some kind of like sorting based method. I know there's, yeah. Again, Miltenyi has a really cute machine that I want really bad that'll do this for you. Yeah. Yeah. No. Hey, thanks. I'm really good. No, I don't know. I'm telling you, brain, they just come out easily. Do you work with, yeah, you work with brain, right? No?
Yeah. But in our lab, we're using like fat cells.
Oh, adipocytes. Uh-huh. Wait, so a fat cells, like adipocytes?
Yeah.
Oh, yeah, it depends. Again, like, so yeah, this is skeletal muscle. Heart was bad. It was really messy. Really? Big?
So it becomes the whole.
I just feel like with droplet-based, right? Like, it's Split-seq or another.
Possibly, but then the cells just connect first.
Okay.
Whoa. Did not know about that cell type being a pain. That's crazy. Maybe that's why we don't see any of these
Yeah.
ones.
Yeah, the oil top, the oil won't mix.
Yeah. That is cool. Yeah, so each experiment is gonna have like, I don't know, do brain. Brain is great. Hopefully your PI is like brain. Okay, then I just have kind of a summary of what the entire workflow will be. Yeah, you're preparing your single cell or single nucleus suspension. It's going to take forever, probably, unless you're going from a cell line. If you're going from a cell line, I'm jealous of you. Now, I used to do that. You barcode your cells, so it's either going to be the, I'm not going to talk about plate, it's going to be the droplet-based method, where you're streaming your bead barcodes in with your cells or nuclei, and I get your nanodroplets. Oh, I missed this. Oh, wait, one of the most, another hard part about preparing single cell or single nucleus. It has to be the right concentration. You have to count. And the counting is, you think it's easier, or you think it'd be easy, but it's not. Again, due to debris, like your automatic cell counter will count debris as cells, and you'll get some number that's completely off. So I really recommend fact sorting, or this is what I do. I do hemocytometer all day, every day. Same with DAPI, see what's a nucleus, count only the nuclei. Okay, what are they? Yeah, either droplet-based, or you split and put your cells into plates containing their specific barcodes. You reverse transcribe your RNA to cDNA. That's either going to happen in droplet or in the cell or nucleus itself. You release the barcoded cDNA, and then you do your, where did I see this? Oh, sorry. I should say you do first strand synthesis. Oh, no, sorry, you already did. Okay, then you do cleanup. Okay, you do bead-based cleanup, which, have you all know what bead-based cleanup is? That is a huge part of all these protocols. It actually takes a surprising amount of the time each time. Just waiting for things to bind, waiting for things to I don't know, incubate. Your ethanol washes, takes forever. Then you do your template switching to get your second strand of your cDNA. You clean up your cDNA again with the beads, and you do your QC check of your cDNA using a bioanalyzer and a qubit to measure your concentration. So this is a nice hippocampus cDNA sample that I made the other day. We're expecting a pretty high size distribution, considering you're supposed to be getting like the full length transcripts, more or less. Let's see. Then for short read sequencing, you have to tagment, right? Illumina can only sequence fragments of a certain size. So yeah, you do your tagmentation. You amplify your tagmented cDNA. You clean up again. You do another bioanalyzer QC check and qubit concentration measurement. And you expect this nice clean peak for your library, which we have. Yay, brain. And yeah, and then you go for sequencing. And then this is my last slide. That's just comparing the different sequencing platforms, just keeping in mind that you're going to have, this is another thing to take into account, I guess, that depending on which platform you choose, probably this one or this one, you're going to have a different number of reads. So a different number of reads per cell at the end of the day. So yeah, that's it. Oh, good. Okay. Any questions? What kind of literature? Like how did I mean, yeah, for the Split-seq, yeah, there was this paper that was our lab got excited about, and then we contacted the company and asked the company a million questions and stuff like that. I mean, it depends on what you're interested in. Like if you, yeah, like you just want a broad overview.
Yeah, well,
Probably there's papers.
well, if you were looking for like, I work with mRNA, and I'm looking for transcriptomic changes, and N6A-seq is like a thing. I don't know anything. I'm like a second person. I have no like this one. So if you were looking for transcriptomic changes, but I'm using a cell line right now.
Great.
So
Okay.
what would you suggest if I was looking for? I have like a target in mind.
Okay.
But I don't know how to approach.
So you mean like a target, like a specific gene? Like how many?
Autophagy as a process, so all the genes like involved in autophagy. So there's like a list.
Okay, because I was gonna say if it was like just a few genes, honestly, like
Yeah.
probably be useful because
Yeah.
it's cheaper.
Yeah,
But-
no, I was gonna say qPCR. Yeah. Is there a reason, like, are you gonna move from the cell line to something else? Is there a reason to
Yeah.
try to
And
capture
so I
like,
go into these maps.
oh, okay.
So, I start in like neuronal like cells.
Uh-huh.
And then I have scRNA and like knockdown to try to look at some targets because I'm basically pulling together several different concepts and trying to find like a mainstay. Like that's what my thesis is supposed to be like. So seeing if they have correlation to each other. So I'm affecting like M6A methylation and then looking downstream for genetic changes because it's going from RNA to like changing the transcriptome, but then that transcriptome possibly affects the protein. So I really think it's a thing, but I have to try to convince my PI it's a thing. So I'm trying to like look for a method that RNA-seq is what she wants, but then there's spin.
Well, yeah, no, definitely start. I mean, RNA-seq would be good. I mean, again, like if you're only looking for a few genes, qPCR is not
If you thought it was an argument
the worst.
Because I don't know if you guys know Ty Quinn in Leslie Thompson's lab, but he did RNA-seq and he suggested qPCR to start, but then she wanted to do RNA-seq, but she'd rather, she's like real maintenance. She don't want too many hands on it or whatever. She'd rather send it off to an expert. So what if you were like looking and you were trying to find some changes? And I think it's other things too. Like she likes to do RNA-seq because we tend to go back and look to see what other genes may be involved because we have 30 billion different things.
Yeah.
So I think I really like the split-seq you were mentioning. You said it's high, it's lower throughput, but more thorough.
The one that would be that one would be like more plate-based, like you're fact sorting an individual cell into each well. Yeah.
If they're neuronal-like but not neurons, they should be okay for now. But when I go to neurons, then it's not practical. So they don't. You were saying that you need isolation due to markers, right? You isolate things possible.
Yeah, I mean, you could do that still, all that still with bulk RNA-seq. Are you specific?
Like individual cell type
Yes.
populations? Okay, then yeah, you definitely want to do single cell.
Mhm.
Or like isolate cell type populations that are predefined by markers beforehand.
Yeah, but a lot of the stuff I need is cytoplasmic. Like all MCA has a lot of. It's looking like localization. Sorry.
So guys, no, good. This is exactly the kind of conversation that we want to have.
That's fun.
We can have this over lunch.
Yeah.
I feel like this is a lunch conversation.
Yeah.
You know? So does anybody have other questions? Or